A pleasant good morning to everyone who are gathered here for this guest lecture on modernization of plant breeding and plant health management. It gives me a pleasure to welcome our chief guest, Dr. B.M. Prasanna, Director, Global Maize Program, International Maize and Wheat Improvement Center, shortly called as SIMET, which is based at Nairobi, Kenya. Actually, he was invited as a chief guest in the International Millet Conference and Futuristic Food Expo organized by our university in honor of International Year of Millet 2023 and Centennial Year of Millet Research. Yesterday, he has also signed a memorandum of understanding with our university. At that juncture, he has discussed about many opportunities for involvement of our university in the 1CGIAR and Plant Health Initiative programs on maize. The director of CIMET has been invited by our Vice Chancellor to present a lecture on this occasion for the benefit of students and also faculty members. We are very grateful to have Dr. Prasanna here for graciously agreeing for this guest lecture. I feel extremely honored to introduce our distinguished chief guest who is occupying a prestigious position in an international organization. Dr. Prasanna is an experienced professional in the field of genetics, molecular breeding, and genomics. He was born in Bapatla, Guntur in 1965. He earned his undergraduate in Bapatla Agricultural University from 1981 to 85 and attended his postgraduate and doctoral program in Indian Agricultural Research Institute from 1985 to 91. He began working as scientist in 1991 and continued in that position until 96. He was then promoted as senior scale scientist and remained there from 1996 to 2000. And then he became senior scientist in 2000 till 2008. Based on his meritorious contribution in the field of his research, he was recognized as ICR National Fellow for the tenure of 5 years from 2005 to 10. And during this time, he was elevated as Principal Scientist in the Division of Genetics, IRA, New Delhi, during 2008. Later, in 2010, he has occupied this prestigious position of Director, Global Maize Program, SIMET, Nairobi, Kenya, and he is serving there for 13 years till date. And he is the recipient of several awards and honors during his service, like FAO Fellowship at the University of California, USA in 1998, and also ICSA Dr. BC Deb Memorial Award during 2021, ISGPB Dr. Johinder Singh Memorial Award during 2001 to 2, IARA Best Teacher Award during 2004, ICAR National Fellowship during 2005, and NAS Associateship during 2005, IARA BP Paul Memorial Award during 2007, and so on. And Dr. Prasanna has led the CGIAR research program in maize from 2015 till 2021, an alliance of over 300 research and development institutions globally. He has been at the forefront in tackling the challenges of maize lethal necrosis disease in Eastern Africa since 2011, and also the follow-me-worm in Africa since 2016 and in Asia from 2018. The 1CGAIR Plant Health Initiative was created in 2021 by Dr. Prasanna and the larger CGIAR team in collaboration with a wide range of partners throughout the world. I would like to provide an overview of the CGIAR Plant Health Initiative for the benefit of our students and also scientists. This initiative aims to strengthen inter-institutional linkages for effective plant health management, especially in the low and middle income countries like Africa, Asia, and Latin America. It mainly focuses on the monitoring, surveillance, prediction, risk assessment of transboundary pests and pathogens, and implementing IPDM program. This PHI focuses on the wide range of pests and diseases that harms vegetables, potato, sweet potato, cassava, banana, legumes, and cereals like rice, wheat, and maize. Dr. Prasanna is also the current leader of Plant Health Initiative, which includes 80 domestic and international partners in addition to 10 CGIAR program. He has published more than 200 research articles in various international journals of repute and has a Scopus H index of 46 and a Google Scholar H index of 64, I index of 184 with more than 16,000 citations. With this introduction, I would like to officially welcome our main speaker to the podium to give their guest lecture on modernization of plant breeding and plant health management. I hope attending this lecture is a wonderful opportunity for our students. We are eagerly waiting to hear from you, sir.
Good morning to all, both online and offline participant, senior faculty members, pros and biotechnology directors, CPPS, and my students of senior undergrads and also the masters and PhD student from various discipline. So today we have the great speaker and Dr. B.M. Prasanna. Yes, so madam given a elaborate biodata, but other side is he's one of the wonderful teacher. Whenever you ask any IRA students, we ask Prakarna, he's a very wonderful teacher. So it's very difficult to be a teacher and a scientist, but he's being a big role, dual role. He will do that. So he's when yesterday evening our vice chancellor asked, please address our students. Immediately he agreed because he's so passionate to talk to the students and he's doing more than 40, 50 lectures throughout the world and he's always the man on wheel. Every time today he's in Asia and maybe tomorrow, maybe another day is in the Europe. So such a dynamic personality. And also whenever you see Maize literature, whenever see anything on molecular breeding, the Dr. Prasanna paper is the first paper people refer. Even when I come to Maize after a period of moving from millets and whenever I read the Prasanna paper because he's any new ventures, he is the first person to address the issue. Even recently, when all over the world the Maize program is struggled with fall armyworm, when he was the global Maize director on the Maize breeding program, he immediately took up the challenge and he complete line on tolerance to fall armyworm. So whenever you refer again Maize fall armyworm story, resistant breeding, when you can refer on the Dr. Prasanna paper. That's why any scientist when work on a particular crop, you will always new challenges come, whether lethal necrosis or fall armyworm or the nutritional trait or the land races. Wherever you touch the area, you can read the Prasanna paper. Just type Maruti Prasanna, you will get all the information. Such a wonderful speaker is today, and the man in global lead on the many programs. Recently, the plant health management, and really we are fortunate to be here to hear your lecture. And also our students are very fortunate to that morning to have clarification on, because whenever any maize breeding program or many soil plant health management, if you have doubt on that, definitely you can refer Prasanna. And also he is very eager to take students and in the international program. So whatever the doubt on that, whether the international program you want to work or the scientific area you want to get clarification, after his lecture you can get a clarification. So thank you for come over here and TNAU and be with us. I welcome Dr. Prasanna.
Good morning, friends. It's a great pleasure to be here, especially in front of the students. And both Dr. Shanti and Senthil have been very kind in showering appreciation. I am deeply grateful. In fact, in 1991, my career started in Coimbatore. I was 6 months at the Sugarcane Breeding Institute, Coimbatore, when I joined as a scientist, and that was one of the wonderful times I spent in my life with my wife, who is now the head of the Department of Microbiology at IARI, and my very young son at that time. My first son was 1 year old. So lots of memories of Coimbatore. Okay, today I will talk about two aspects. One, about modernization of crop breeding programs. And the second is how plant health management issues, what are the issues that are emerging and what do we need to do in order to tackle the challenges of plant health threats through an integrated approach. For the students, it is very important to understand what are the major drivers and the underlying factors that are challenging food and nutritional security. Uh, there is a publication that came up in 2021 from FAO. This is called The State of Food Security and Nutrition in the World. And this is a wonderful publication to refer to. And there they highlight five major factors that are, or four major factors that are challenging the food and nutritional security. The first one is climate variability and extremes. Climate change is indeed a reality. It affects in a huge way, diverse spheres of our life. The second is the ongoing conflicts. Conflicts are there everywhere between countries, within a country. And these conflicts also do affect in a major way the food security of millions of people. Then the third is economic slowdowns and downtowns, including the effect of devastating pandemics like COVID-19. And the fourth one is that we talk about healthy diets. Healthy and balanced diets are extremely important. But what is a fact is that most of those balanced diets, healthy diets, are also quite unaffordable to people who survive on very meager incomes. For rich people, for middle class people, it is still affordable. But a lot of poor people cannot really afford to have healthy diets. So these are the four major issues that they highlight and deal with them in a very detailed way. When it comes to climate change, these are the two major regions, sub-Saharan Africa, which is this one, and this part is the Sahel region. It's a dry desert. So sub-Saharan Africa means this particular area. And South Asia, these are indeed the hotspots for climate-induced variabilities and extremes. Most of the food security is affected due to the changes that are happening in these two regions, which are also home to billions of people. Okay, so and people who survive on less than $2 per day are predominant in these regions. So they are deeply affected by climate variabilities. Yet at the same time, what is the solution? We need to nurture diversity. Genetic diversity is indeed critical if you have to tackle climate change. Without diversity within a crop, without diversity across crops, crop breeding cannot progress. So if you look at why I am showing this picture is I'm passionate about maize diversity, which is one of the best in the world. If you look at the diversity of maize in Mexico, if you have a chance in your life, go to Mexico and go to Oaxaca, go to Puebla, go to Jalisco provinces. You will see some of the best diversity. Do you know what is the name of this beautiful landmass of Mexico? This is world's longest ear maize. It's 45 centimeters long, as much as this. Every year, farmers have a competition. Which farmer has grown in that year the longest ear maize called Hala maize? It's pronounced as Hala. It is written as Jala because J in Spanish is Ha. So it's Hala maize, 45 centimeters long, restricted to certain belts of Mexico. And it's not present anywhere in the world because of the volcanic soils. It's extremely productive. And similarly, there are others like Tuxpanos. There are purple mazes. With the purple maize, they make tortillas or chapatis, and that is highly rich in antioxidants, and it is as good as anything else. It's very much preferred by many sections of people in even in US right now. If you go to Texas and Florida, it is very highly preferred. So purple maize, white maize, yellow maize. So in CIMMYT gene bank, We have as many as 28,000 maize landrace accessions. Each landrace accession is diverse within itself. 24,000 of them have come from Mexico alone. So Mexico is the country, the farmers where they have domesticated maize, and that from there it went to all over the world. Today, maize is one of the most widely grown cereals in the world, and the production has reached almost 1 billion metric tons. It's the highest producing crop in the world. And therefore, there is no, and if you come to Africa, not just in Mexico, for Mexicans, maize is life. Everything around their world, including food, including customs, religions, everything revolves around maize. Similar to our culture, rice in this part of the world is just like that. Okay. So in Africa, if you go 40 million hectares of maize, maize is an introduced crop there, but maize consumers are so many. 300 million people in Africa eat maize on a daily basis. It's called ugali in Kenya. It's a white porridge. Just like we eat rice with others, they eat ugali with other food components. So maize is not just a feed crop that propels the poultry industry. Maize is a food crop for millions of people in Latin America, Africa, and in some parts of Asia too. Around 20 to 25% of maize produced in Asia is consumed as food. When I was ICR National Fellow, I had an opportunity to collect land races of maize in India. If you want to collect the best land races of maize in India, you have to visit northeastern Himalayas. And within the northeastern Himalayas in Sikkim, every family, every farmer grows maize, and they hang it in their houses like this. Every farmer. And this is a special land race in Sikkim called Sikkim Primitive. Each node, there will be a year. There are 9 years, 6 to 9 years on a single plant. That means there is no apical dominance. Apical dominance prevents not more than formation of 1 or 2 years. It's a physiological phenomenon. There are genes within maize that suppress apical dominance and leading to ear formation in each ear, in each node. So I had an opportunity to characterize all these land races when I was at IRA and as ICR National Fellow, including all these diverse land races. This is called Putli Makai. Putli means butterfly. These are transposable elements. So the transposable elements have given these color patterns. So local people call it Putli Makai. This is called Gadbade Makai. That means something Gadbad has happened. That means white has been contaminated with purple. So there are all sorts of maize. That's why Pools Henry Poincare gave this wonderful statement. It says the scientist does not study nature because it is useful. He or she studies it because he or she delights in it. And he or she delights in it because it is beautiful. If nature were not beautiful, it would not be worth knowing. And if nature were not worth knowing, life would not be worth living. What a fantastic statement. If nature were not beautiful, it would not be worth knowing. And if nature were not worth knowing, life would not be worth living. What a profound statement, right? My dear young students, that's diversity. That's diversity. Understanding diversity, capturing the diversity, analyzing this diversity, utilizing this diversity enriches your life. Genetics and plant breeding, therefore, revolves around that diversity. And that is the treasure trove to meet all present and future challenges. So now coming to the crop breeding pillars. These are the crop breeding pillars. We need to understand what is environmental adaptation. How do you characterize the phenotypes? How do we understand the genetic diversity and use it? and what kind of genetic information we can generate in our breeding programs. So these are the four pillars. If you have to be a good geneticist, you need to think constantly about these four factors. These are the pillars. And you have to always ask the question, how strong is your network? Are your protocols and processes well optimized? How efficient is your data generation, data curation, data analytics? Students need to be very strong in this area. If you have to be successful as a scientist, capacity to collect data, analyze data, and present this data in an appealing manner is extremely critical. One of my uh, teachers used to say, he used to visit, he was a DDG of ICIR when I was a scientist. He used to go all around the country. He is very fond of me, of all the work that I do. He used to go to every scientist whenever he used to give a lecture in crop science or horticulture and ask, do you know Prasanna? Usse dosti karo. He used to say, Usse dosti karo. That means make friendship with him. What he is indirectly telling them is network, network, network. You cannot achieve anything in life alone. Network. So that is the important principle of life. If you walk alone, you walk a few miles. If you walk together with your friends, you walk very far. So these three questions are very important. Irrespective of whatever you do, you may be a pathologist, entomologist, geneticist, breeder. The ability to accurately phenotype will always remain important, no matter what evolutions it may take in terms of breeding technologies. Editing today, yesterday it could be transgenics. Sometime after 10 years, there could be something else. But remember, the importance of phenotyping will never vanish. Phenotyping is king. If you are good at phenotyping, you are kings or queens. And the phenotyping, what do we mean by phenotyping? Phenotyping means the capacity to unravel genetic differences through morphology or through phenotypic traits. The capacity to unravel those genetic differences consistently and robustly. That means today you don't get one data and tomorrow after some time you get another data that is not consistency or robustness. That in genetic terms we call heritability. Heritability of trait expression. That means if your heritability is high, no matter how many replicates you put, the phenotype will more or less remain the same. That is heritability. No matter how many locations you may evaluate a genotype, the phenotype will remain consistent. That is heritability. And nowadays in genetics, we don't use simply CV, the coefficient of variation. We use the statistical parameter called heritability. How many of you are aware about heritabilities and how to calculate heritabilities? Please raise your hands, the students, my dear young students. Okay, undergraduates, you may not know, but postgraduate students must know, must. And you must use this parameter in describing all your experiments, not simply presenting mean, range, and CV. Okay, very important. If you don't know, talk to your quantitative geneticists or biometrical geneticists and understand the concept. The second is you must develop protocols, whether it is for drought stress, heat stress, low nitrogen stress, waterlogging stress. Detailed protocols need to be generated by breeding institutions, and these need to be disseminated widely so that not only you, your partners also follow consistently the same protocol and get data which is comparable. Otherwise, you'll be comparing apples with oranges, which is not really good. So precision and consistency in trait evaluation. They are very critical. In addition, now in the last 5 to 6 years, there has been emphasis on high throughput. That means not just collecting data for endlessly for weeks and months. How soon you can collect precise data within a matter of a day, or within a matter of few days, or even within a matter of few minutes. This is called high throughput phenotyping. And this high throughput phenotyping in case of maize, I have a brilliant scientist by name Mainasara Zamanallah. He is from West Africa, fantastic guy, young guy, who optimized the processes for proximal and remote sensing of maize phenotypes from our drought stress field block. He analyzes it and he gets the data, empirical data, within a matter of few minutes, instead of a technician walking from plot to plot to plot to plot and taking hundreds of minutes for recording data. Similarly, this can also be applied for disease resistance. This is maize streak virus. You look at the plots, different genotypes. Some are showing excellent resistance, some are moderate, some have succumbed. So getting that kind of disease data. Similarly, tar spot complex is another major disease in Latin America. We use multispectral imaging again to differentiate a susceptible and a resistant genotype through drones. The other way of doing it without drones is called proximal sensing. Proximal sensing is, for example, ear imaging. By taking a picture, put the ears there, take a image, and then process that image without having to shell the ears. This can be applied for many crops. Proximal sensing and remote sensing. And some companies have even gone on, for example, when you harvest maize or wheat, it already records data. Can you believe it? That is called combine. Combine is running, it is harvesting ears. The ears go through a sensor, and the sensor already collects data of what is potential protein content, lipid content, oil content, everything right when the harvesting is done. And the record goes to a computer. Look at the technology. This is called NIRS analysis, near infrared spectroscopy analysis on a combine itself at the time of harvest. Plus the laboratory analysis of samples for certain traits. Earlier, we used to take several days to analyze amino acid levels. Nowadays, we don't have to. Wet lab chemistry has changed significantly too. There are high throughput procedures even for analyzing nutritional quality traits. So therefore, at multiple levels, proximal sensing, imaging, NIRS analysis on the combine, laboratory analysis of samples, all the spectrum has changed. Students need to be trained in those, right? If you want to become a modern breeder, and if you want to enter an industry, talk about phenotyping confidently, you need to be well aware of this. Gone are those days where laborious data collection, manual data collection happens in evolved breeding programs like ours. We don't go through such. Even data collection is not through handbooks. It's all digitized. You have barcodes on your genotypes. You just scan the barcode and then enter the data. Boom, it gets recorded. All our technicians are trained on barcoding and recording data on tablets. No hand analysis. Now another revolution is DNA-based markers. DNA-based markers also have evolved. Can you believe it has been almost 4 decades, 40 years since the first DNA marker was discovered by these two gentlemen, Eric Lander and David Botstein. They both were brilliant human geneticists, and they discovered RFLPs way back in 1980s. The first paper got published in American Journal of Human Genetics in 1980. So it has been 43 years. They established RFLP maps, linkage maps for the first time in humans, and then went into discovery of human genome sequence. Eric Lander was the guy who led the human genome sequencing. Okay, and then a number of platforms have emerged: low density platforms, medium density platforms, high density platforms. So nowadays we don't even do analysis in the labs because they are excellent commercial platforms which can generate the data, boom, for much less cost than what you can do in a lab. Within a matter of few days, they can analyze your data. You have to simply send your lyophilized leaf samples or whatever, and they will, and then tell these are the markers I want to analyze for. I have Intertech, LGC, which can generate the data and send me data within few days. Then I use this data to select plants, which plants to go for and which plants not to go for. So there are platforms now available. Using such platforms, now you can convert this beautiful collection of your germplasm in the gene bank, not just phenotypic evaluation. You can use now markers to analyze what beneficial alleles are there in different land races. Why does a Hala maize look like a Hala maize? What actually made that Hala maize so long? Yeah. So this is called seeds of discovery. We had this project from almost 2010, funded by the Mexican government, and we comprehensively characterized the landrace accessions in our gene bank and created what we call Maize Molecular Atlas. Not only understanding where they are coming from, which part of Latin America each accession came from, what is their passport data, but also what is their genotypic data and phenotypic data. Genotyping and molecular breeding need not be always marker assisted backcrossing to convert a disease susceptible line into a resistant line, or one with no natural nutritious quality into a better quality line. That is one approach, marker assisted backcrossing. And through that, you are alleviating one or two of the defects that are present in widely adopted lines. But if you want to make progress in breeding, marker assisted backcrossing is not necessarily the only option. Because with marker assisted backcrossing, what are you doing? You are taking a line which is 10-year-old or 15-year-old, or taking a variety which is 10-year-old or a 15-year-old, and then converting that. That means your basic genetic background is remaining 95 to 98% same. Only you are adding one trait or two traits. That is useful under certain circumstances, but not always good for making progress in breeding programs, because your genetics basically is 10-year-old, 15-year-old. That's why we use in CIMIT what we call marker assisted forward breeding. Forward breeding means when you do breeding, during your breeding scheme, right within F2, you make selections using markers. Take those selected plants, move forward. That means markers become integral part of your breeding scheme itself. Every plant is analyzed genotypically. Every plant then is selected on the basis of markers. This is forward breeding. And you can do that for specific traits for which you have breeder ready molecular markers. For those which you do not have breeder ready markers, you should do for complex traits like drought, waterlogging, or things like that. Then there are no specific one or two or three or four genes. You have to select the whole genome. And that is what we call whole genome selection or genomic prediction. That means you analyze the genotype information of all your lines. You collect phenotypes for half of those lines. And for the rest of those lines, you predict their performance based on their relatedness to those lines for which you have both genotypic and phenotypic data. That is called genomic prediction. And that genomic prediction is very important for making progress on complex quantitative traits. How many PG students are here? Can you raise your hands? Very good. Are you well aware about genomic selection and or prediction? You are taught? Have you done? Have you read papers? है ना? पढ़ा है ना? बाद में questions पूछूंगा. Okay, I'm just kidding. But what is important is the markers is not only for generating information on simple oligogenic traits, but markers can be used powerfully for genomic prediction. The third important application of markers is quality assessment, quality control. This is called QA/QC. QA/QC is quality assessment or quality control. During a seed production chain, you will have breeder seed, foundation seed or basic seed, then commercial or certified seed, right? You know that, right? That is seed value chain. Breeder seed is produced by the breeder in small quantities. Then you scale it up to a reasonable size. It's the basic seed or foundation seed, which is disseminated to partners. Then the partner uses this basic seed to produce commercial seed or certified seed or truthfully labeled seed. And then he sends it to farming communities through agro dealers. At different steps in the seed value chain, in whichever crop you may be working on, there could be possibility of impurity happening, genetic impurity or contamination happening. So markers could be very powerful tools to do quality assessment, quality control, QA/QC. In our seed, in maize, we have a set of 100 SNP markers, single nucleotide polymorphism mark. We published even a manual on how to do QA/QC in maize using molecular markers. Not only we do that, but we train our partners, national programs and seed companies on QA/QC so that they can use it. They don't have to have laboratories. They simply need to fix the leaves and send it to a commercial service provider. And the commercial service provider already knows what markers to use. And he or she analyzes the data and sends the data back. Looking at that SNP data, you can then understand how much homozygosity is there, how much heterozygosity is there, and is this completely a contaminated seed lot so that you can take immediate action. So that is another powerful application of marker. QTL profiling can be done for the new lines, whether a new line is having a nutritionist quality or not, or whether it is new line is having disease resistance or not. So that is called QTL profiling. Genomic selection, I already talked about. You can make 30 to 40% saving in time and cost for a breeding pipeline. 30 to 40% saving by not phenotyping everything, but genotyping all. Genotyping is very less costly. Phenotyping is more costly if you have to generate data from multiple locations. So this is genomic prediction. It's an integral part of each and every breeding pipeline of mine, whether it is in Latin America or in sub-Saharan Africa or in Asia. Another most important truth, no product from my program gets released without having marker analysis, marker-based breeding at some stage or other. So there is no difference between conventional breeding and molecular breeding here. Molecular markers are integral part of your whole breeding strategy. When we were students, me or Senthil, there used to be two schools of thought. Some breeders said, oh, we are conventional breeders. What will molecular markers do? Nothing. Molecular markers, molecular breeders used to feel special too, somebody like me. But now there is nothing like that. If you don't do molecular markers in your breeding program, you are not a breeder. Simple. If you don't have that knowledge or expertise, should you do? What should you do? Dosti karo. Dosti karo with that fellow or with that girl who has expertise in that. But there is no excuse, right? Dosti karo. Remember that. Not just for going to the movie or enjoying in a party. Okay? Dosti karo every day. Then once you do analysis at different stages on the station in TNAU or in Adudurai or in some other curriculum, you generate phenotypes. That's not enough. You need to also analyze the performance of those materials. This is called stage 5. Stage 1, 2, 3 is the breeder's funnel. Then selected materials from this go to extensive on-station trials in my program across different countries. Once this on-station, these are all controlled by scientists. Then from that, we select a subset of materials which has to be analyzed in hundreds of farmers' locations. That's called stage 5. Not under scientist management. We just select farmers. Okay, these farmers are in relevant market segments, and those farmers under their own management condition, they evaluate those subset of genotypes and we collect the data from them. Why do we collect? So when we select the farmers, we also make sure that at least 40 to 50% of the farmers are women farmers. Women farmers. This is very important because not all male and women farmers have same way of looking at performance of your materials. Women value certain traits differently compared to men farmers. Okay, for example, quality is very important for them. Processing is extremely important for them because they have to feed the children. They're bothered about their nutrition. So in our breeding programs, therefore, we need to have deliberate selection of women farmers, collect their data, understand their patterns of selection, understand what they value the most, and you have to value their selection. You have to value if your products have to be successful. So the concept of breeding is not gender neutral. Breeding is gender responsive. Breeding has to be gender responsive. If you have to be a very good commercial breeder. So that's what we do. This is just an example in Eastern and Southern Africa where we evaluate each year in over 700 farmers' fields our products and collect the data. This gives us also opportunity to understand G by E by M, genotype by environment by management under farmers' conditions. Gender disaggregated preferences for products and traits. Incorporate more robust data. Then when we do the final announcement of products, we take not only on-station performance data, we also take on-farm performance data and then identify these are the 3 or 4 best products in that particular pipeline in that particular year, and then we announce to the partners. CIMIT is a not-for-profit organization. We don't register varieties or produce seed and commercialize seed. We give them these to a wide array of national partners as well as seed companies. And then they go to the market with those products. Friends, if you have to do again successful breeding, you need to be aware of data management and decision support system. Yesterday, I had a conversation with colleagues, including the Honorable Vice Chancellor, about this. Breeding is about data compliance, standardization, analysis, efficiency. Data management is quality control, technology transfer, or progress, data visualization. If I type in my system, give me a white, early maturing maize line with these resistance, then in my breeding, in the analysis system, it should pop up. That is called data visualization. Tell me which particular lines have this particular haplotype with disease resistance. It quickly screens my germplasm and identifies those materials which have that haplotype. This is called decision support system. And that decision support system has to be built in a platform. And that platform has to accept data from diverse sources, pedigree data, phenotypic data, and genotypic data, automate routine data analytics and query process, visualization, what markers to use, what lines to use in breeding process, what models to use for genomic selection, how do you predict performance of progeny based on plant process prediction. So this is the evolution I would like to see in India. Not even a single institution, single breeding institution in India has this capacity at present. We say, Mera Bharat Mahan hai. That pride is always good. We must have the pride in other spheres. But we should constantly learn and accept, right? If we don't recognize where we are not good, how will you make progress? No way. If I keep on telling CIMIT is great, CIMIT is great, CIMIT is great, that doesn't work. If I have to be a good leader, I need to know what others are doing good. Where are they better? And then I need to improve my program. That's what we need to do. The next big thing is deep learning or machine learning. It's a controversial subject because it can be used for both good and bad. It can, based upon your patterns of Googling and patterns of many things, Google can identify what this person likes. Because you are constantly ordering online, it can predict your pattern of behavior. So artificial intelligence is already in use, already in use. The important thing is artificial intelligence can be used beneficially in breeding programs. You can do, you can predict phenotypes. You can do prediction of genomic values, disease forecasting, fertilizer recommendations based upon the soil health quality, weather-based risk analysis, yield prediction. All these things can be done using machine learning algorithms. All these things, they are already being done. Again, this is an area where we need to strengthen our curriculum, our capacity and understanding of, even faculty members need to be trained, students need to be trained. But there is nothing like one particular machine learning algorithm that can do this for all possible applications. Time series analysis, geographically weighted ML or machine learning with local interpretability, graph-based neural networks. So there are different applications for which you require different kind of models or algorithms. So a lot of bioinformatics work, people who have very strong understanding of data coding will become part of breeding strategies. This is a reality. हम लोग बायोलॉजी में आए इसलिए है कि मैथमेटिक्स से हमको डर है. 90% of people hated mathematics. Therefore, they took up agriculture. No, I'm just kidding. Some of you are very good. Not all of you. Okay. But there is nothing like now statistics, quantitative genetics is very much part of breeding. So you need to get trained. So there are excellent articles coming up on this. We need to mechanize or modernize our breeding operations, various levels, not just tractors for the sake of it. There are combine harvesters, which can, and there are combines with data analytic capacities. Drip irrigation, every farm of ours, there is nothing like furrow irrigation or sprinkler irrigation. Our drought blocks, everything, optimal blocks are all drip irrigated so that we save water, huge quantities of water. Then we use these seed dryers. These are medium-sized seed dryers where you put your harvest in these boxes, and then underneath there will be perforated holes. You can control the temperature, and within a matter of 3 to 4 hours, it can bring down the moisture content from 35% to 12%. And you don't have to wait up to physiological maturity to harvest. You can harvest your material 2 weeks or 3 weeks in advance and bring down the moisture level quickly. Select your next set of kernel seeds and then plant them in advance quickly. That means what you are doing 1 season or 2 seasons a year, you are creating 3 to 4 seasons in a single year. This is accelerated breeding. Speed breeding does not mean only creating big facilities to phenotype. This is also a part of speed breeding. Accelerated seed drying. Then digitalizing seed systems. Entire seed system can be digitalized. You can have excellent cold store and you can digitalize your entire seed inventory. So process of using digital technologies to transform business processes and operations with the goal of improving efficiency, reducing cost, enhancing quality is called digitalization. So this can be done for multiple reasons: better data, improved traceability, better access of your seed, assured quality, increased productivity, targeted reach, and many others. So yesterday we talked about this too with the Vice Chancellor about opportunities to digitalize our seed systems, not just in maize or wheat or rice. In any crop, you can do it. And this can be much, much better for your this thing. Then strategic partnerships for private sector on strategic areas, for example, BT maize or genome editing, phenotyping platforms. For this, we created huge networks. We have 45 seed companies across 4 countries in South Asia, which are partners in a consortium. This is called International Maize Improvement Consortium. You can look here, 87 members are there in this consortium. So with them, we feel the pulse of the market. We meet their germplasm needs. We get their sites for expanding our phenotyping network, and we provided value-added breeding services to them, including marker analysis, double haploid development, and so on. Then follow me worm. We created this huge screen houses when this follow me worm called Spodoptera frugipada. It's a very devastating polyphagous pest. It can the moth can migrate almost 500 kilometers during its lifespan. 500 kilometers, such a powerful flyer. Some moths have reached even 1,000 kilometers. Can you believe it? 1,000 kilometers they are flying, attacking maize. If they don't find food there, they fly. They go to a new maize field or a sorghum field and then eat away that. So how do we tackle such a devastating transboundary disease? We created this network of screen houses. Not only this country, it's an air-conditioned city. There are no fans, no ACs anywhere in my office or at home. Nowhere. No ACs. Constantly 20 to 25 degrees Celsius throughout the year. Can you believe people think Africa matlab bahut garmi hai? But India is more garmi. When I come to India, I feel the pain. Okay, so our misperceptions. Okay, this is another such complex that we created in ICRISAT. So that what we are doing here is that thousands of genotypes are tested every year under artificial infestation of fall armyworm. We mass rear the insect, we generate the neonate larvae, and the neonate larvae are used to infect each and every plant at the V5 stage, and then at one more infestation at the R1 stage, and then you assess the behavior of each genotype. This facility exists nowhere in the world. 16 screen houses, 16, not just one or two, so that we can analyze hundreds or thousands of genotypes. Through that, we identified, because we have this diversity in our germplasm, tuxpenos, Caribbean flints, offer resistance to many insect pests. We evaluated those, we identified fall armyworm tolerant inbred lines. We shared with 92 institutions in 34 countries globally since 2018. So even for a devastating pest, you can find genetic variation in maize. That's how powerful maize, the diversity is. You want to read more, there is a paper that I published in 2022 on host plant resistance to fall armyworm management. What is the relevance and status and prospects in Africa and Asia? Look at the phenotype here. The tolerant inbred line under no choice trials. No choice means even within each greenhouse, there is a partition of half this size here. You can grow 40 rows of maize within that partition, only one entry, one germplasm entry, and artificially infest all the 40 rows. In another chamber, you have next genotype. In another one, the no choice means the pest has no choice. Either you eat and die, or you devastate a susceptible chuck like this. This is a foolproof analysis. That means there is no escape. In a choice experiment where A is there, B is there, C is there, D is there, the insect has a choice. If not this, you don't like this, you go to the next one. But in a no choice, that is the litmus test. You understand? So this is very, very important in evaluation of germplasm for insect pest resistance. Using this technique and revalidating the responses, we then, you see, not only evaluate the plant behavior, but also the yield. Here you can see a lot of black spots because the fall armyworm can bore through the ears, eat the developing kernels. Whereas in a resistant genotype, it doesn't like that. So not only the yield is higher, but the ears are clean. So these hybrids have been already released in Kenya, Zambia, South Sudan, and Malawi in the last 1.5 years. And several other countries are testing it. This is something that we would like TNAU to do. Come up with fall armyworm tolerant hybrids. Use our germplasm. Get it done, evaluate them. You don't have to establish 16 screen houses. Establish even 2 or 3 that will serve the purpose. Then genetics and breeding will continue to evolve. The choice is in front of us. Either you keep doing the same, or occasional improvement, or continuous improvement, with innovation if you have to make rapid progress. So students, be well versed with the best possible use of new tools and approaches to study genetics, to increase genetic gains, and for developing and deploying impactful products. Partnerships. Whether you are a MSc student, PhD student, or a scientist, partnerships. They are critical for impacts. But breeding alone will not work. One thing is to increase genetic gains. Another thing is to protect the genetic gains from devastating diseases that are happening. That's why we call eco-friendly plant health management. This is important to build resilient agri-food system, eco-friendly plant health management. Despite so much of published literature on the consequences of hazardous pesticides, this is a drastic reminder to us it has no reduction. Pesticides have in fact increased in their consumption. Many countries have increased consumption, unfortunately. And the impact of this increased levels of pesticide is much greater on women farmers. Women. In many African countries, man's job is simply drink and sleep below a tree. The woman is actually doing all farm operations, bichari. She is doing plowing, she is doing weeding, she is carrying children on her back and applying pesticide without even personal protection equipment. Can you believe it? This is the fate. This is the fate. And when they apply these pesticides, which are cheaper and they are duped by agro dealers, they damage their health. They damage the health of their children. They damage ecology. So, friends, plant health management is so critical. You have to save environment. You have to save people. You have to save crops. That's why I keep saying healthy crops are vital for a healthy planet. If you don't have healthy crops, there is no healthy planet. That's the reason why we established this plant health initiative. And this plant health initiative is with 10 CGR centers, with ECPA, CABI, WorldVeg. Madam partners with WorldVeg. So she is indirectly or directly contributed to the plant health initiative. And similarly, over 120 non-CGR partners distributed globally. I led the formulation of this entire initiative. I am not just a geneticist or a breeder now. I expanded my horizons. Okay, that's what life has to teach you. Even at 54, 55, you can learn. You can do many things. You can demonstrate your value. When MLN broke out, when fall armyworm broke out, I did dosti with people, published some brilliant manuals. I demonstrated how to tackle these diseases. So CGIR therefore did not recognize my discipline. They said Prasanna is the guy who should lead this initiative. I'm so proud of that. So I wear multiple hats now. I'm the director of Global Maize Program of CIMIT. I'm the 1CJR Global Maize Breeding Lead. I'm member of the leadership team of CIMIT. I'm also the CJR Plant Health Initiative lead. No time to sleep. 18 to 20 hours per day work. 18 to 20 hours. When I can work at 57, imagine what you can do. Okay, right? You can learn so many things, read, learn. So one of the first objective is to create this diagnostics and surveillance networks. Using our CGR germplasm health units, we linked up the national plant protection organizations in several countries globally, in Mexico, in Colombia, in Peru, in Eastern and Southern Africa and Western Central Africa, in South and Southeast Asia. Then we are undertaking surveillance on several pests and diseases in 7 crops for different pests and diseases in collaboration with the national partners. We have created image-based and molecular detection tools now so that we can make the process of diagnostics and surveillance much more rapid. Tumani app is one such one. LAMP is a diagnostic assay. So several image-based and molecular detection tools. Then not only molecular diagnostics or surveillance, we also need to create risk analysis, preparedness for rapid response against this wide range of pests and diseases, banana bunchy top, southern rice black streak dwarf virus, which happened in India last year, potato purple top, zebra crop chip disease, fall armyworm, maize lethal necrosis, potato leaf blight, yam mosaic virus. So name the crop and the major disease or pest. We are now collecting data on the emerging risks, models for risk prediction, and data for decision support. This is another work package. Work package 1 is on diagnostics and surveillance. Work package 2 is on risk assessment, data management, and guiding preparedness for rapid response in low and middle income countries. Work package 3 is integrated pest and disease management. Not just using fall armyworm tolerant hybrids. Fall armyworm tolerant hybrids plus biological control or biopesticides or intercropping with relevant crops, agroecological management. This is called integrated pest and disease management. Improving the knowledge base of the farmers, collective actions, public-private partnerships, and providing visibility and political voice for IPM. This is what Madam is doing now in the Dharmapuri district. And this is based on World West success in identifying a tomato package in Andhra Pradesh against fruit borer and Thoromia absoluta, replacing 11 to 14 sprays of chemical pesticide with no compromise on marketable yield. So this model is now being partnered with the TNAU on for participatory demonstration trials in Coimbatore and Dharmapuri districts. So TNAU is actually already playing an important role. What we would like to do is to expand beyond tomato IPM in the years to come. You have the capacity. You have excellent human resources in crop protection. And therefore, you are in a very good shape. We created what we call innovation platforms, plant health innovation platforms. We can create one such innovation platform in TNAU. The purpose is not to demonstrate one particular protection measure against a pest or disease, but do experiments on how different protection measures can be combined. Those protection measures need not come only from your university. It could be a university with another public research institution, or it could be a university with private sector. So what we did, this is Kibako in Kenya. We established for fall armyworm a plant health innovation platform. We invite hundreds of farmers, and those farmers then evaluate each IPM package and rate them. I like this. This is looking fantastic. I don't like this treatment. We collect that data besides scientists' data. This is participatory engagement. So the purpose of an innovation platform is to bring different partners on the same platform, evaluate IPM packages, then based on cost benefit analysis and efficacy, identify which packages are best and then scale them up. This can be done, right, in Tamil Nadu? So that is what we would like to get in TNAU, an innovation platform, plant health innovation platform. Regional plant health gender networks. See, I'm glad to see so many girls sitting here. We need to engage with the women farmers, make them understand that gender and social inclusion is very vital for improving plant health management. We need to educate them. We need to provide them with relevant knowledge. We need to make them partners in co-creation of IPM packages. So we have established in Southeast Asia with 50 participants from 8 countries already in 2022. This is a regional plant health gender research network. So we can do that in South Asia. Can TNAU take the lead for that? Okay, that's another opportunity that I see here. You need to facilitate digitalization and labor-saving technologies for women and young farmers. Include those farmers in pest and disease detection and early warning. Identify incentives for women and young farmers in adopting plant health innovations. social inclusion in our scaling approaches. So we can provide you with gender integrated research tools, gender checklists for inclusive interventions. We can partner with you on how to do this better. So this is another most important aspect. If you want to learn more about this, there is a publication we brought out. If you type CGR Initiative on Plant Health, you will see a technical report that we got in 2022. So go to that and understand what all work we are doing. I just scratched a tip of the iceberg here. Okay, with that, I will stop now and take questions from you. Thank you so much. I was a teacher for 20 years. I taught 4 courses in genetics at IRI. When student ask, do not ask questions, then I consider it as a failure. Okay, so ask questions. Any question, there is no question that is not relevant. Especially undergrad students. Let's start with those undergrad students. Any question you have? Then we'll go to MSc, then go to PhD.
Morning, sir.
Good morning.
It's a very informative and very we are very grateful for this lecture. Yeah, I wanted to just ask like a little bit. I wanted to know about this no choice trial. Can you just bit explain what is that?
Which
No choice trial.
no choice trials. Because in a normal breeding experiment, you evaluate side by side several genotypes, right? Like this room, you have different genotypes. You put them in the same. Imagine this is a screen house, okay, and you artificially infest all of them. Then there will be insect. It is biological resistance, but it is simply non-preference. So you need to understand the difference between non-preference and native resistance. Native resistance through metabolite production or other means that this particular genotype has innate capacity to resist a insect attack. Non-preference means I just taste a few leaves. I don't like this, or I like this lesser, but this one is much more palatable, so I will go there. So in a choice experiment, there could be confusion. So initially you do the choice experiment, you identify good genotypes, then you go for no choice trials. No choice trials means there is no several genotypes in the same screen house. There will be partitions. There is a big screen house with 3 or 4 partitions. In each chamber, then you have only one genotype. Then also you do the complete infestation. Then if the insect, if genotype is susceptible, it will be finished. If the genotype is resistant, the insect takes it, doesn't like it, or tries to feed on it, but there is no choice and it will die because it doesn't feed. So that is the difference between choice and no choice. No choice is a very good indication that there is a native resistance within that genotype. Okay, it's not a breeder's variety, it is a farmer's variety. Because you see, maize has been domesticated by the Mexican farmers since 10,000 years back. 10,000 years back, maize was first domesticated by Mexican farmers. What is domestication? There is a wild relative called teosinte. From the teosinte, the modern maize has originated. So what farmers did through multiple generations is selected plants which have got certain properties, natural mutations. So the single stalk ear has become a multi-road ear. Okay, what is, so there are a number of traits that changed over the process of domesticating teosinte into modern maize. And once modern maize came up, several land races have originated in different ecologies. So again, farmers, by keeping on selecting for something, they keep on selecting for year length. It might have taken 100 years, 200 years, but in that process, a new land race emerged called Hala maize. Similarly, a purple maize is a selection by the farmers. So like that, farmers have selected for diverse land races, which are still grown even after hundreds and hundreds of years. They are still grown in Mexico. Farmers grow them. So that is not a institution bred variety, but it is a natural land race.
Told us that there will be like even for knowing who's growing biggest halamis, is it in Mexico or where?
What is it?
Usually there will be like even to know who is growing the longest maize, Mexico.
Similar to our basmati rice. Why is basmati rice naturally so rich in those jasmonates and other flavonoid compounds or aromatic compounds in particular belts? It is because of the soil properties. That is genotyped by environment interaction. So whether it is land races or sometimes even modern varieties, certain properties emerge in certain belts, whether it is Darjeeling tea or whether it is something geographic appellation, they call it indication. So certain traits are particularly fixed in certain this thing because of various climatic and edaphic factors.
So you talked about no choice trials. How we are tackling the soil microbiome impact on the plant, like in resistance and susceptible cell.
Soil microbiome has influence on some of the traits. Definitely there is influence on some traits, but not particularly with regard to traits like fallamiworm resistance. Fallamiworm resistance, there is no association found so far with soil microbiomes and capacity of a genotype to resist fallamiworm. So it doesn't really matter. So but soil microbiome is important for certain other properties. For example, there are associations found for abiotic stress tolerance in maize and in many other croplands. There is association between soil microbiome and yield levels in certain traits, in certain genotypes, in rice, for example. So soil, my wife is a microbiologist. If I don't go back praising microbiomes, then I'm finished. Okay. So, but that is a joke, but basically it's very, very important. Soil microbiome, we need to understand better how microbiomes influence. Even the gut microbiome, there are excellent papers, human gut microbiomes. It can influence your anxiety, your depressive behavior, your happiness, your health. There is a lot of understanding about human gut microbiome and the relevance for integrated medicine. Okay, so don't, that's why people say don't just keep on eating medicines or pills on your own prescription, without prescription. What do? They affect your gut microbiome. And that has influence on your health. So just on the counter prescriptions, one should never use just like that or doses exceeding what the doctors recommend. The reason is very simple. It's about your gut microbiome.
Can we know about going research projects on AI-based detection of plant diseases in your institute, artificial intelligence-based?
Yes, absolutely. There are several institutions working on artificial intelligence based. For example, in our plant health initiative itself, there is artificial intelligence based plant disease analysis. So I can refer you to Michael Selvaraj. Michael Selvaraj is a product of TNA. Okay, you must be knowing him very well. And he is at C at Columbia. He is an excellent guy. I call him Sachin Tendulkar. He's just this height. But excellent scientist. Excellent scientist. And he is working on artificial intelligence based disease diagnostic apps. Okay. m.selvaraj at cgir.org. Okay. GM crops are not panacea for all our yields. Okay. It is one of the tools. I'm not fascinated only by the power of any technology, but we need to do a judicious analysis of which particular methodology is appropriate for what specific trait and under what socio-economic context. Okay. If you develop a very powerful innovation, and if that innovation is not socially suitable because it is so costly, it is unaffordable, or there are so many other factors that influence the power of adoption of a technology, then its limit will be experienced. All said and done, GM Cotton has made tremendous strides in the country. You believe it or not, GM is the technology that has seen the most rapid growth than any technology in agriculture within a short span of time. From 1996, if you trace the growth of GM crops in the world, it is one technology which has the most wide adoption. No doubt about it. But if you look at Africa, maize is Africa is around 50. They have been struggling to introduce some BT maize because CIMIT does not work on generating transgenic technology. No. Our 90% of our focus is on breeding. Conventional plus molecular. As I said, there is nothing like conventional and molecular. Breeding itself is so powerful. I showed palamiworm tolerant hybrids you can get, which are excellent. And the advantage of these palamiworm tolerant hybrids is they have polygenic resistance. Polygenic resistance. That means multiple genes are controlling the resistance. In case of BT, in certain systems in US, it is completely BT maize. There is nothing, hardly 5% is normal maize. So in certain contexts, certain countries, in certain traits, GM has worked. Not for everything. Nothing like drought tolerant transgenic maize. Very powerful. All the progress that we made in Africa, Asia, Latin America, all conventional. 7.5 million hectares are grown with IR. 5 million hectares. Conventional breeding or normal breeding process without transgenic technology has made huge impact too. But when there is no option at all, then you go to transgenic. Traits are there in the market, transgenics, insect resistance, herbicide resistance, some nutrition quality trait. That's it. How many crops? Then half a dozen. Bayer or Corteva or Syngenta to develop transgenics for all the crops, they will not do. Full reason. For growing finger millet, pearl millet, so many crops. Like only on GM technology. It will never happen because the technology is not right? Part of legal. So when CIMIT itself, when we decided, we are a powerful breeding center. Said our strength is what? Germplasm bank. The strength is what? Where can we make impact? Thousands and millions of farmers breeding. But we will partner with Corteva or Bayer. We have to introduce all. Genetic background remains natural. Simple. Got my point? So my request to you is develop a balanced thing. So completely no anti. It should be context based. Each technology has got its merits and merits. Don't blindly say that transgenics are the best, and don't blindly say that transgenics is बेकार है, करना ही नहीं चाहिए. That debate endless. People are polarized because of activists. I wrote a book way back in 2000 on transgenics. 2000, almost 23 years back. precisely emphasizing that it has to be need based, it has to be context based, not about be blindly opposing or blindly defending any technology. Got it? So many technologies will come. Have a careful and balanced perspective. Understand what are their strengths, what are their weaknesses, who is controlling the technology, what is accessible, what is affordable, where do we need to grow what? Do okay? Simple.
Dr. M. Prasanna, Director of CIMIT, our Director CPPS, Dr. Shanti Madam, the other faculty members, as well as student friends. Very good morning to all. Indeed, a very great pleasure. It was a vote of thanks on behalf of our Tamil Nadu Agriculture University. We are much grateful to our Vice Chancellor Madam for providing an opportunity to have an MOU with CIMIT, as well as to have a very enlightened guest lecture on this modernization of plant breeding and plant health management by Dr. Prasanna. So we are very much thankful to you, sir, for enlightening us with your talk for the students as well as for the faculty members. So as per your talk, surely we will have a friendship with you for the for the research collaboration as well as for if anything possible for the exchange of students as well as other important facilities. And for the information of house as well as for the so we are having a very good facility in bioinformatics with a good server and other for data analysis. Surely we can have a collaboration with you on this big data analysis. Thank you. Thank you once again for the wonderful lecture and for the enlightening our students. I'm also much thankful to our Dean School of Postgraduate Studies for motivating us to organize this lecture within a very short period of time. And thank you very much, sir. And also, we are very much thankful to our Shanti Madam for the elaborate welcome address and to all the faculty members who have attended this lectures, as well as for the student friends, got some motivation from his lecture. Thank you, Onanda. So we request our director.